very closely related to mitosis and also very closely spelled to mitosis is our next topic, meiosis. And this, for one, is a huge difference, but we'll get there. Before I start explaining the process of meiosis, there's a couple terms that you need to be familiar with. The first one that we'll talk about is called a homologous chromosome. It's where you have two sets of a chromosome, one coming from mom and one coming from dad. So, as you can see here in this picture down here, these are all human chromosomes here. And you notice you have one that will come from your mom or your dad, and the other that will come from your mom. Okay, so, and the exception is our sex chromosome. So here's another just example to show you. So right here in this picture, we have an unduplicated chromosome. Okay, here we have a duplicated or replicated chromosome, or a sister chromatid. And then here we have a pair of sister chromatids that belong to the same thing, so that'd be like having this and this. Whoops, if those two. That is a homologous pair. One comes from mom, one comes from dad. The only exception is our sex chromosomes, because you either have an X or two X's or an X and a Y. So two sets of chromosomes, homologous pairs. Another important vocab term is diploid. So this is a cell that has both sets of chromosomes. So we are all diploid organisms. Okay, di means two, and they're giving N for the number of chromosomes. So meaning we have 2N because we have one from mom and one from dad, giving us 46 total, or 23 pairs. So the opposite of diploid is haploid, which is a cell that only has one set of chromosomes, or a single set of chromosomes, and its symbol is N, and you should know both of their symbols. And you should know examples. Like examples of a haploid cell is called a gamete. A gamete is sperm, eggs, or pollen. Okay, so those are all haploid cells as well. They each have 23 chromosomes, and they have 23 chromosomes because you don't want to have too much genetic information. So these gametes and haploid cells are waiting to be fertilized in order to create a diploid organism or an adult organism. So just to show you, here is a karyotype of a haploid. See how it just has one set of each chromosome? Okay, versus a diploid, which has two sets of each chromosome. And hey, look, gamete is following us around the page. Cool. Alright, so since we're all made up of diploid cells, we have to, in some way, take our cells and half the information in them in order to get to a haploid cell. And we do this by a process called meiosis. Okay, so it cuts the number of chromosomes in half per cell. It's referred to as sexual reproduction because it produces different varieties. And it produces four genetically different haploid daughter cells. And there's two parts, meiosis I and meiosis II. Alright, meiosis I. The purpose of meiosis I is to separate the homologous chromosomes. And if you remember, here we go, homologous chromosomes, we're trying to separate those from one another. So we're going to take these two and put them in their own cell, and take these two and put them in their own cell. Alright, so interphase 1 is much like interphase of mitosis. DNA has been replicated. So just like in the karyotype that you saw, we have one from mom, one chromosome from dad. So in interphase, the DNA is replicated, and now we have our homologous chromosomes. After interphase comes prophase. So prophase, we're going to talk about two different things in it. One thing you have to know is the tetrad. Okay, so tetra means four, and basically, again, these chromosomes are going to condense into these X shapes, but also they're going to line up into a row of four, just like this, over here, which is called a tetrad. Alright, the much more important part of prophase 1 And the cool, like, set aside from mitosis part of meiosis is in meiosis, there's this event called crossing over that happens. So as these homologous chromosomes pair up in their tetrad, see that over here? Sometimes their little feetsies get crossed. Oh my goodness. And then they end up with different feetsies, and that produces new combinations. So crossing over is exchanging parts of the chromosome to produce new combinations. And just to reiterate, you see that over here we have homologous chromosomes, one from dad, paternal, one from mom, maternal. Alright, so just like in meiosis, metaphase comes right after prophase, except we call this Metaphase I because there's two parts to meiosis. So again, very similar, in Metaphase I, we have our spindle fibers that will attach to the homologous chromosomes, and the chromosomes you see are still lining up in the middle of the cell. Anaphase I of meiosis, just like Anaphase I of mitosis almost, except we're talking about homologous chromosomes instead of sister chromatids. So you see here our homologous chromosomes are being pulled apart. Telophase I in cytokinesis, we have our nuclear membranes reforming. Okay, our cells beginning to split. If you're talking about an animal cell, here we have the cleavage furrow. Alright, and we have two haploid daughter cells. So just two cells so far. Brief review, meiosis I separates homologous chromosomes. So here we have our interphase, we have our chromosomes replicating, DNA replicating, and then over here, what's that called? Look at the little feetsies crossing over. Okay, so again, metaphase lining up in the middle, and then we have our homologous chromosomes separating or being pulled apart in anaphase. Meiosis II, its purpose is to separate now these sister chromatids. Okay, there's no interphase 2 needed because the DNA does not need to replicate. So it'll pretty much uncondense and then condense right back in prophase 2. And then it'll go into metaphase 2. Metaphase 2, very much the same, lines up in the center. This meiosis II looks almost identical to mitosis. Anaphase 2, the sister chromatids are being pulled apart. Telophase 2 in cytokinesis, so we're having a cell split, and then we end up with 4 haploid daughter cells. Alright, so here's what meiosis II looks like. Remember, its purpose is to separate the sister chromatids, or these X shapes. Okay, so you notice we have the same thing going on in 2 cells now. Alright, so our prophase, we have our condensed chromosomes. Metaphase, they're lining up in the middle. Anaphase, sister chromatids are being pulled apart. Okay, and telophase 2 in cytokinesis, the cells are splitting, and we're going to end up with 1, 2, 3, 4 daughter cells, haploid daughter cells. Alright, so here we have a diagram of meiosis I and meiosis II. Here we start over here with interphase 1, if you will. But we have our DNA being replicated in there in a growth phase. And then we go this way to prophase 1, where we have our crossing over occurring between our homologous chromosomes. Then we have our metaphase 1, where they're lining up in the middle. Anaphase 1, homologous chromosomes being pulled apart. Telophase 1 in cytokinesis. We have our cells splitting, okay? Now we have 2 cells going on, so we're going down here. Now we're going to move in this direction. Alright, so we go right into our prophase 2. Look, our sister chromatids are now lined up in the middle. This is metaphase 2. Our sister chromatids are being pulled apart here. This is anaphase 2. And here we have our telophase 2 and cytokinesis. So we have our 2 cells splitting into 4. And look, oh yay, now we have 4 haploid daughter cells. So since they're haploid, remember they have the N. Overview of meiosis I, meiosis II. Meiosis I, the purpose, to separate the homologous pairs. Okay, purpose of meiosis II. Now we have sister chromatids in each cell. We're separating the sister chromatids. And now we're going to have 1, 2, 3, 4 haploid daughter cells. See how it has half the amount of information down here than what was up here. Diploid, haploid, haploid, diploid. Comparing mitosis and meiosis. So you notice that they are starting out with the same amount of information, 2N. They're both diploid organisms. Okay, except here we still end up with diploid organisms with the same amount of information that we started with here. But over here we end up with half the information that we started with, and this information is different. You see how this has a blue foot attached to the red thing? Okay, so we get 4 haploid daughter cells. We have 2 identical diploid daughter cells in mitosis. Alright, now you got to watch this cool animation of meiosis that'll review some key terms for you.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The 2 homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in 2. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.